Hello students, welcome back to my channel. In this video, I am going to give you a brief lecture on photorespiration. So photorespiration is a light dependent process occurs in plants. So where the plants are consuming oxygen and they are releasing CO2. Okay, photorespiration have so many alternative names. Let's see one by one. It is also called as oxidative photosynthetic carbon cycle. So why? Because here RUBP is going to fix the oxygen. That's why it is an oxidative process. Whereas, so it is also a photosynthetic process. Why? Because here the RUBP acts as a substrate. That's why it is a photosynthetic process. So it is also a carbon cycle. Okay, so the second alternative name is nothing but glycolate pathway. It is also called as glycolate pathway. Why? Because glycolate is the primary substrate for photorespiration. Okay, so glycolate formed as an intermediate compound in this cycle, which is a primary substrate. That's why this cycle is also called as glycolate pathway. This is also called as C2 cycle. So why? Because the glycolate, which is a primary substrate formed in the photorespiration, that is a 2-carbon compound. That's why this cycle is also called as a C2 cycle. So actually the photorespiration occurs with the coordination of 3 organelles. They are chloroplast, peroxisomes, and mitochondria. These 3 organelles, they work collectively together for the C2 cycle. Okay, let's see the C2 cycle in detail. So photorespiration generally reported in C3 plants due to the dual nature of RUBISCO enzyme. So let's see what is RUBISCO, which is an abundant enzyme responsible for the photosynthesis. It fixes the atmospheric carbon dioxide, we know that. So RUBISCO nothing but ribulose bisphosphate carboxylase oxygenase. So it has the dual nature. If it acts as a carboxylase, it fixes the atmospheric CO2. If it acts as an oxygenase, it fixes the atmospheric oxygen. So that is the dual nature. Due to the dual nature of RUBISCO enzyme, there will be occurrence of photorespiration. Okay. The dual nature of RUBISCO enzyme was reported by 2 scientists. They are Orgen and George Bauss. These 2 scientists have reported the dual nature or dual function of the RUBISCO enzyme. Generally, RUBISCO enzyme prefers to fix the atmospheric carbon dioxide. But the conditions are if the conditions are different, like if the oxygen concentration in the environment is high and the temperatures are high, during high oxygen concentration and high temperatures, it prefers to fix the oxygen. So instead of taking carbon dioxide, it prefers to fix the oxygen. So that is the dual nature. Okay. So due to this, photorespiration occurs. Okay. Let's see the pathway in detail. Photorespiration occurs by the coordination of three cell organelles. They are nothing but chloroplast, peroxisome and mitochondria. They collectively work for this cycle. Okay. So what happens in the first reaction? Let's see. So instead of taking carbon dioxide, RUBP accepts oxygen in presence of an enzyme RUBISCO, resulting in the formation of 3-phosphoglyceric acid and 2-phosphoglyceric acid. 3-phosphoglyceric acid immediately undergoes Calvin cycle. Okay, so 2-phosphoglyceric acid is involved in the cycle. So that is the first step. In the second step, the 2-PZA is undergoes dephosphorylation, resulting in the formation of glycolate. That is the second step. So let's see the third step. This glycolate will diffuses into the peroxisome. There it converts into glyoxylic acid. Glycolate undergoes oxidation. So here oxygen is added to the glycolate. So it undergoes oxidation. So then it converts into glyoxylic acid plus H2O2 is released here. So actually H2O2 is toxic to plant cells. That's why immediately it should be broken down into H2O plus nascent oxygen. So in presence of an enzyme peroxidase that is present in the peroxisome, which converts hydrogen peroxide to H2O plus oxygen. So let's see the next step. In the next step, glutamic acid is added to the glyoxylic acid. Then it converts into glycine plus oxaloglutaric acid. So glycine enters into the mitochondria. Okay, let's see the next step. So in mitochondria, 2 glycine amino acids, they combine together, resulting in the formation of 1 serine. In this reaction, ammonia and carbon dioxide both are released in the mitochondria. So the release of carbon dioxide in the mitochondria is nothing but, that's why this is called as photooxidation. Okay. So in mitochondria CO2 is released, that's why this is also called as photooxidation. This step is called as photooxidation. Okay. So now the serine again diffuses into the peroxisome. There it converts into hydroxypyruvate. Serine is combined with glyoxylate in presence of an enzyme aminotransferase. There it converts into hydroxypyruvate plus glycerin. Okay. So now hydroxypyruvate undergoes reduction reaction. There it converts into glyceric acid. So now the glyceric acid is again entered into the chloroplast. So the glyceric acid undergoes phosphorylation. It converts into 3-PZA. So that is also utilized in the Calvin cycle. So photorespiration occurs with a combination of 3 organelles. So let's see the steps in detail. Okay students, these are the steps involved in photorespiration. Let's see one by one. So in the first step, the first step and second steps occur in chloroplast. There oxygen is added to the RUBP with the help of an enzyme RUBISCO oxygenase. So there it converts into RUBP converts into 3-PZA and 2-PZA, 3-phosphoglyceric acid and 2-phosphoglyceric acid. The second step also occurs in chloroplast only. There the 2-PZA is converts into glycolate in presence of an enzyme phosphorylase. Here one water molecule is added and one inorganic phosphate is given out. So let's see the next third step, that is glycolate is entered into the next organelle, that is peroxisome. That's why 3 and 4th steps occur in peroxisomes. Okay, so glycolate undergoes oxidation reaction in presence of an enzyme oxidase. It converts into glyoxylate. So in this step, hydrogen peroxide is released. So actually hydrogen peroxide is toxic to the plants. So plant cells. That's why immediately it splits into water and oxygen with the help of an enzyme catalase that is present in the peroxisome itself. Let's see the fourth step that is also occurs in peroxisome. Here the glycolate and glutamic acid combine together, resulting in the formation of glycine and oxaloglutarate in presence of an enzyme aminotransferase. Okay, now the glycine enters into the mitochondria. There two glycine amino acids, they combine together with the help of an enzyme transferase, resulting in the formation of another amino acid that is nothing but serine. In this step, carbon dioxide is released out and ammonia also released. Due to the release of carbon dioxide in mitochondria, this step is called as photosynthetic oxidation. Let's see the next two steps. They occur in again peroxisome. So serine enters into the peroxisome. There it combines with glyoxylate in presence of an enzyme aminotransferase. There it converts into hydroxypyruvate plus 1 glycerin also released here. Okay. So next step also occurs in peroxisome. There hydroxypyruvate undergoes reduction reaction. Okay. So in presence of an enzyme reductase, hydroxypyruvate is converts into glyceric acid. Here one NADPH is utilized. It converts into NAD+. Okay. Now the last step that occurs in the chloroplast. Okay. So glyceric acid released in the peroxisome, which is enters into the chloroplast, which diffuses into the chloroplast. There it undergoes phosphorylation in presence of an enzyme phosphorylase. It converts into 3-PCA. This 3-PCA also utilized in the Calvin cycle for the preparation of sugars. Okay. So okay students, this is about photophosphorylation. So let's see what are the consequences, how they overcome photorespiration. Let's see. Actually, photorespiration is a wasteful process. Why? Because it decreases the photosynthetic efficiency. Okay. So and also it wastes energy and carbon also. That's why it is a wasteful process. To overcome photorespiration loss, C4 plants and CAM plants are evolved. In case of CAM plants and C4 plants, so CO2 is accumulated in the cells, so that facilitates the carboxylation reactions only. In case of bundle sheath cells, there is a release of CO2 in the bundle sheath cell itself, so that promotes the carboxylase activity of the RUBISCO. So in case of CAM plants, there in the daytime CO2 is released in the mesophyll cells. So that concentration of CO2 in the mesophyll cells, that facilitates the carboxylation reactions only. Okay. So in this way they avoid, C4 plants and CAM plants, they avoid photorespiration loss. Okay. This is about photorespiration students. Thank you for watching.